Dr. D, Dr. D, Dr. D. Dr. D, Dr. D, Dr. D. Dr. D, Dr. D, Dr. D. Explain stuff.
Hey everyone, Dr. D here, and in this video we are going to be doing a Exam 1 review for Biology 2420 for Tarrant County College. This is from our Cowan book, 6th edition. So let's go ahead and get started. Recall that this exam will cover chapters 1, 3, 4, and 5. So let's get started with chapter 1 first. So obviously, please know any terms in bold. The exam will be directly from these lecture handouts, the material that we covered in the lecture. So obviously here you see in bold microbiology. Know the definition for that and know what is studied by microbiologists, right? These are the major groups of microorganisms. And you should know one thing in particular is that viruses and prions are not alive, right? These are small infectious agents, whereas bacteria, fungi, protozoa, and helminths, they are organisms. They are alive. Sorry, I'm just skipping forward to see other important material for you to know. You should know that the eukaryotes have a true nucleus, while the prokaryotes, which include bacteria and archaea, they do not have a nucleus. Okay. Okay, here we are. Now, what you should know is that bacteria and archaea are two domains of life which are prokaryotic. They are much smaller than eukaryotic cells, 10 to 50 times smaller. They lack membrane-bound organelles, such as the nucleus, endoplasmic reticulum, etc. And they typically, remember, they only have one chromosome, and it's a circularized chromosome. And that chromosome is called the nucleoid when it's referenced as inside of a cell. You should know the term parasites. You should know that it was Louis Pasteur who disproved the concept of spontaneous generation through his elegant swan neck experiment. Robert Hooke was the first to take pictures of microorganisms like fungi and publish it in scientific journals. We should know that Anthony van Leeuwenhoek, he was one of the first pioneers of the microscope with his pinhole microscope. We should know about Koch, Robert Koch, who linked specific microorganisms with specific diseases through his postulates. Remember, we talked about Koch's 4 postulates. Dr. Wendell Holmes, remember, he was the first to observe that counter to what you would expect, mothers who gave birth at home experienced fewer infections than mothers who gave birth in the hospital. This is what led Dr. Ignaz Semmelweis to hypothesize that washing hands and sanitation may have an effect, right? So he was the one who really promoted hand washing and saw better outcomes there. We also should know about this pioneer, Joseph Lister. He was the one who first utilized hand washing and sterilization of surgical instruments such as scalpel and other surgical instruments during surgeries, which led to reduced infection, post-operative infections. We should know that Pasteur also invented pasteurization when he studied fermentation and how the process of fermentation works. Koch, through his studies with linking diseases with different bacteria, he helped to establish a series of proofs verifying germ theory of disease. Do you remember the concept of binomial nomenclature, or the binomial system, where the genus name is always capitalized while the species name is lowercase? And then remember that this name needs to be italicized or underlined. So for example, if we're talking about Homo sapiens, we need to capitalize the first word and lowercase the second word, and then we need to either italicize the whole thing or underline the whole thing. If you're writing it by hand, you underline it. If you're typing it out, it's preferred to italicize it. And do you recall we talked about taxonomy? These are levels of classifications of life. Remember, there are 3 domains of life: archaea, bacteria, and eukarya. And then within those domains are different kingdoms, and then different phyla, class, order, family, genus, and species. So please know these levels of taxonomy, this hierarchy of taxonomy in order, right? So if I said, for example, what's missing, and I leave out class, and there's a list like domain, kingdom, phylum, blank, order, then you know it's class, for instance, right? So please know these levels of taxonomy, the hierarchy of taxonomy in order. And then you remember where we talked about the 3 domains of life: bacteria, archaea, and eukarya. And I told you something fascinating, and that is that archaea and eukarya share a more common ancestor than either does with bacteria. So please know that archaea and eukarya are more closely related than either is to bacteria. And this is, against what you might think, is because bacteria and archaea are both prokaryotic, so you would assume that they're very closely related, but that's not the case. Right? So those were the important parts of chapter 1. Hopefully that made some sense. Let's take a little break with Gizmo and Wicket, and then we'll hop right into chapter 3. All right, welcome back from break time with Gizmo and Wicket. Let's go ahead and continue on with our Exam 1 review with Chapter 3, which covers tools of the laboratory. One thing you should know for the exam for sure are the 5 I's of microbiology. So please recall these 5 I's. And I'm not going to spend a lot of time going through all these definitions, but like I mentioned before, please know all of these terms. If it's in bold, it's definitely expected that you know it. So please know all these terms like incubation, what's a colony. Remember that it's one bacteria that results in one colony on a plate. You should know that there are different media, and media are classified based on their physical state, chemical composition, and functional purpose. Obviously, you should know these terms here, like what is chemically defined media or synthetic. Remember, that's when we know exactly what's in the media. Whereas you should know that a complex media is not chemically defined, and we do not know the exact makeup of the media, such as an extract. If we extract some nutrients from yeast or beef, we don't know exactly what's in that, right? So extracts they make complex media. You should know these terms here: general purpose media versus enriched media, selective, differential. Remember, selective media select against certain types of bacteria and for other types, whereas differential allow you to distinguish between different colonies that do grow or different bacteria that do grow. And enriched media have some special added nutrient. And again, if it's in bold, like liquid media, semi-solid media, solid media, agar, please learn these terms. Anything in bold should be definitely considered for the exam. Okay, defined synthetic media, complex media, general purpose media, enriched media. And you should know also, look at this, that fastidious bacteria require enriched media, because fastidious bacteria, these are the ones that require special nutrients for growth. All right. Here you can see the difference between a selective media versus a differential media. Now, obviously, you don't have to go through and memorize these giant tables of different types of media and all the different purpose for these media. You should just know the general concept of these different types of media, right? Okay, you should know that blood agar is a type of differential media, and this allows you to distinguish different types of bacteria from one another. So it's a differential type of media. Some bacteria can form hemolysins. These are enzymes that break down red blood cells. And depending on how well these blood cells are broken down, we can determine if a bacteria exhibits beta hemolysis, which is a complete lysis of red blood cells, alpha hemolysis, which is an incomplete lysis of red blood cells, or gamma hemolysis, which means no breakdown of red blood cells. So here you can see gamma hemolysis versus alpha versus beta. Notice that beta, you have this halo, you have this clearing where there's no blood anymore. The red blood cells have been completely lysed near the bacterial colonies, whereas look near alpha, it's not a complete hemolysis. Instead, you have this bruising effect. Okay, know what reducing media is, carbohydrate fermentation media. What is a colony? Remember that a colony consists of just one species, and that's because a colony starts with a single parent cell, which has divided exponentially with binary fission. And so two distinct colonies represent two different species. If the colonies look different, like this white colony over here and this red colony over here, that would be suggesting of two different species of bacteria. Okay. We should know that viruses are too small to see with the light microscope. You need to use an electron microscope in order to see viruses. However, bacteria, archaea, and other forms of life can be seen with our light microscopes. Remember magnification, you should know that term. How many times a specimen appears larger than it truly is. We should know the term refraction, the difference between a simple microscope and a compound like microscope. We should know the parts of the microscope. Do you recall the ocular, which magnifies the image, the objective lenses, which magnify the image, the stage? You should know that the condenser focuses a cone of light onto the specimen. You should know that the diaphragm or iris limits the amount of light through the condenser and helps to improve contrast if you limit the light. You should know the coarse adjustment knob, fine adjustment knob. Okay, please know these terms: magnification, resolution, and contrast. Remember, magnification is how many times larger a specimen appears than it truly is. Resolution, you can think of that as a level of detail, how close two small objects can get without appearing as a single object, and contrast, how well your specimen stands out against the background. Okay. You should know about the resolving power of the lenses. And by the way, remember your objective lenses. You have a 4x scanning objective lens, a 10x low power objective lens, a 40x high power objective lens, and a 100x, I don't know why it says 200x here, it's a 100x oil immersion lens. And do you remember how to get total magnification? Remember, to get total magnification, you multiply the ocular lens, which always has a 10x magnification, with the particular objective lens that you're using. So if you're using the 4x lens, then your total magnification is 4 times 10, 40x. If you're using the 100x lens, objective lens, then your total magnification is 100 times 10, or 1000x. Again, I told you what resolution is. You should know what resolving power is. Remember, we talked about the electromagnetic spectrum, and I told you that red light has a longer wavelength than violet light. And you should know that the shorter the wavelength of light, the more energy and the more resolving power. So the shorter the wavelength of the energy that you're using, the better the resolution will be, and the more energy that particular light has. Okay, I told you about the oil immersion lens, and I told you that you have to use oil, you have to use a drop of oil when you're using the oil immersion lens, the 100x objective. And the reason for that is to prevent the scattering of light rays, to increase numerical aperture. What you're doing when you add oil, you should know that you're reducing refraction, that means bending of light, and you're reducing reflection, right? By adding oil, you are gathering more light into the objective lens by reducing refraction and reflection of that light. I told you what contrast means. Okay, we should know the different types of light microscope: bright field, dark field, phase contrast, and interference. And what I want you to know is this: bright field is your standard kind of compound light microscope, the type of microscopy that we've been using in the lab this semester, whereas dark field produces a dark background and a bright sample. You should know that dark field produces a dark background and a light sample. This helps you with contrast. Phase contrast, it uses light tricks. It uses a phase ring, which makes your sample darker than it really is. So it helps with contrast. Interference, or DIC microscopy, allows for looking at samples in kind of a 3D form. Okay. Another thing we talked about was staining and the importance of staining. The reason we stain is to increase contrast. And do you remember we talked about the two types of dyes used in staining with light microscopes? Basic or cationic dyes and acidic or anionic dyes. What you need to know is that basic dyes, which have a positive charge, a cationic charge means a positive charge, these are attracted to the acidic or negatively charged components on bacterial cell walls. What does that mean? That means that if you want to stain a bacterial cell, you're going to use a basic dye, because basic dyes will stick to the negatively charged bacterial cell. Remember, acidic dyes, like India ink, we would use acidic dyes not to stain the cells, but to stain the background, right? The slide itself will become stained by acidic dyes, leaving the bacteria clear. So this is a type of negative staining. Positive staining is done with basic dyes so that you can see the cells in color. Negative staining is done with acidic dyes so you can see the background with the clear cells. Here you can see a simple positive stain. Notice that we can see the bacterial cells colored here. This was stained, this first image was stained with crystal violet, E. coli that was stained with crystal violet, and on the bottom you have methylene blue stained Trichomonas. Next we talked about differential stains. Do you remember that differential stains use two different colored stains? This allows you to clearly contrast cell types or cells. So there was a gram stain which allowed us to differentiate between gram-positive and gram-negative cells. There was the acid-fast stain which allowed us to distinguish between acid-fast cells and non-acid-fast cells. The endospore stain which allowed us to distinguish between the vegetative mother cell and the little endospores that they can form if they are a member of Clostridium genus or Bacillus genus. And then the capsule stain, which allowed us to see the capsulated bacterium. It's a type of negative stain. So here we can see the gram stain. Remember, the gram stain results in purple colored gram-positive cells and pinkish red colored gram-negative cells. The acid-fast stain results in hot pink or kind of fuchsia colored acid-fast cells, while the blue cells are non-acid fast. And do you remember why cells are acid fast? I told you it's because they have this waxy substance in their cell wall called mycolic acids. Mycolic acids in the cell wall or cell envelope of acid fast cells makes them acid fast. And then remember, members of the genus Clostridium or the genus Bacillus, these bacteria can form endospores, and here you can see red endospores and blue vegetative cells. But if you recall, in the lab we did an endospore stain where I believe we used malachite green, right? And so our endospores were green and our vegetative cells were pink. So they used different dyes in this image from your book than we used in our lab. So don't let that confuse you. In lab, if you recall from lab, we had green endospores and pink vegetative cells. And we also touched on some special stains in the lab and in the lecture. I told you about the capsule stain in particular. Remember the capsule stain? Some bacteria have a capsule in their cell wall. And one thing you should know is that capsules are non-ionic, meaning that neither acidic dyes nor basic dyes will stick to the capsule. So in the capsule stain, two different dyes are used. One dye is basic, and look, one dye is basic and it stains the cells. You see these bacillus-shaped cells? They're all stained with the basic dye. But also an acidic dye is used as well, and the acidic dye stains the glass, the background. So the acidic dye stains the background, the basic dye stains the bacteria. What are these halos then? You see these little halos of no color, colorless halos? Those are the capsules, right? So capsulated bacteria would exhibit these halos, whereas non-capsulated bacteria, well, there wouldn't be any halos. The background would be colored, the bacteria would be colored, but you wouldn't see the little halos around the bacteria themselves. And then do you remember the other type of special stain we covered? Flagellar stain. And this is a special type of staining where the flagella are enlarged by a special staining technique. This allows the flagella to be seen. By doing a staining technique that thickens up the flagella, now we are able to see the flagella under the light microscope. And by the way, this is a peritrichous bacterium. That means that it is surrounded with flagella. And that's it for chapter 3. Not a bad chapter, but interesting nonetheless. Let's take another quick little break time with Gizmo and Wicket and come back strong with chapter 4. All right, welcome back. Let's go ahead and continue on with exam 1 review with chapter 4, bacteria and archaea, the two domains that make up the prokaryotic organisms. Remember that both archaea and bacteria have nucleoid, which means their nuclear material is free in the cytoplasm, whereas eukaryotes have a nucleus, which means that their genetic material is inside of a envelope called the nuclear envelope. And also remember that bacteria and archaea usually have one chromosome that's circularized, whereas eukaryotes have numerous chromosomes that are linear. And remember that bacteria and archaea have no membrane-bound organelles, whereas eukaryotes do. Now, we talked about the properties of life. Bacteria are capable of carrying out all of the necessary activities, properties of life, reproduction, metabolism, nutrient processing. So they are organisms. Remember, this is why viruses are not considered organisms. They are incapable of these necessary properties of life. Remember that bacteria are small. Their average size is 1 micrometer. They are too small to be seen with the naked eye, and they are so small. Remember that we need to use 1000x total magnification in the lab with the microscopes in order to see the little bacterial shapes. And speaking of shapes, remember the coccus bacteria are spherical. There can be oval-shaped bacteria, bean-shaped bacteria, pointed shape. Bacillus, remember, bacillus means rod shape. Filamentous bacteria, these are hyphal shape or thin filaments. And Vibrio are curved bacteria. Remember that pleomorphic bacteria or pleomorphism is no distinct shape. The bacteria that are pleomorphic do not have a particular shape. Now, don't confuse shape with arrangement. Remember, arrangement refers to the grouping of different bacteria. So if you have diplo, that means pairs. So diplococci would be pairs of spherical bacteria. Diplobacilli would be pairs of rod-shaped bacteria. Tetrads are groups of 4. Staphylo means clusters, irregular clusters, kind of like grape-like clusters. Strepto means chains. All right. Remember appendages, we talked about flagella and axial filaments for motility, fimbriae, pili, and nanowires for attachment points or channels. I told you about flagella. The three components, 3 parts of a flagella are the long whip-like filament, the hook, which connects the filament to the basal body, and the third part is the basal body. This is the part of the flagella that's embedded in the cell wall. And remember how flagella work? They work by spinning. They work by spinning, and they have rotation. We need to know flagellar arrangement. Remember, atrichous bacteria have no flagella. Monotrichous have a single flagellum. If it's polar and monotrichous, the flagella is attached to one side. Lopotrichous means bunches or tufts of flagella. Amphitrichous means there is a flagella at each end of the cell, while peritrichous means that the bacteria is essentially surrounded by flagella. Remember we talked about taxis? Chemotaxis means movement in response to a chemical signal. This can be positive with positive chemotaxis. That means the cell is moving in the direction of a favorable stimulus. Whereas negative chemotaxis, remember this was movement away from a repellent or something that's harmful. Phototaxis was movement towards light. This would be particularly important for photosynthetic microorganisms. Remember how flagellar motility works? I told you that bacteria with flagella, they can either run when the flagella is turning counterclockwise or tumble when the flagella switches to a clockwise rotation. This allows the bacteria to change direction. And then remember, we talked about an axial filament. An axial filament is a version of a flagella which is inside of the cell wall. The flagella wraps around the body of the bacterium inside of the cell wall, so it's called an endoflagellum. And this is normally found with spirochetes or spiral-shaped bacterium, and it produces a corkscrew twisting motility, a corkscrew type of motility, where the entire bacterium twists through the mucus and drives itself forward. Okay, then we talked about different types of pilus, fimbria. Remember, a pilus is an external structure, and the sex pilus was the main one that we talked about. This is a hollow tube that some bacterium can form, which can attach to an adjacent bacterium and allow for genetic material to be sent from the donor bacterium to the recipient bacterium. Fimbria, remember we talked about how these are short bristle-like structures on the surface of the cell, which allow the bacterium to stick to surfaces. So here you can see fimbriae. These are short bristle-like proteins which extend from certain bacterium, and they have a tendency to stick to each other and to surfaces. Whereas the pili, remember pili are long protein extensions, and the sex pilus allows for what's called conjugation, for horizontal gene transfer. This means that the bacterium can send genes to the recipient bacterium through the sex pilus, and this is known as a type of horizontal gene transfer where genes are shared. Okay, then we have different types of additional layers on the surface of the cell. The S layer is, you normally find S layers on archaea. Our S layers is a type of cell wall with thousands of copies of a single protein linked together. Then certain bacterium can have a glycocalyx, and I told you there are two forms of glycocalyx, a slime layer or a capsule. Glycocalyces are repeating polysaccharide units, which may or may not include protein components as well. A slime layer is a loosely associated glycocalyx. It helps protect the cell from loss of water and nutrients. It can also aid in attachment of the bacteria to a particular surface. A capsule is more of a tightly associated glycocalyx, much more tightly associated with the cell wall than the slime layer. and it is denser and thicker than a slime layer. Remember, the purpose of the capsule was to help to evade the host's immune system so that white blood cells in your body cannot find capsulated bacteria. They have a tougher time with phagocytosis of these cells. Though the capsule can also help with losing water and with attachment as well to some extent. So here you can see the loosely associated slime layer glycocalyx versus the more tightly associated capsule glycocalyx. And remember, we can see these types of bacterium with a special stain technique for capsule stain. Remember, with the basic dye that stains the bacterium and the acidic dye that stains the background. And here you can see that the capsule, which is non-ionic, is not stained. Okay, again, capsules are formed by many pathogenic bacteria. Remember, pathogenic means disease-causing bacterium. Why are they so good for disease-causing bacterium? Well, because these bacteria evade the host's immune system, the phagocytic white blood cells, which are trying to clear the infection. Also, remember that bacterium can form communities. One type of community is a biofilm, example, plaque on teeth. Here are the steps involved with biofilm formation. Remember, here's the surface. Pretend this is the surface of your teeth. The first colonists arrive and adhere to the surface, usually with fimbria or other surface structures which allow them to attach. Then the bacterium divide and they start secreting specialized biofilm secretions, which are sugar and protein. Other microbes with different species can also attach and start secreting biofilm secretions as well. And this can form a mature community with complex function. Remember, we talked about the cell envelope. The cell envelope includes it can include a cell wall, the plasma membrane, and another outer membrane in gram-negative bacterium. And then you could also have, in addition to all this, you could have a glycocalyx, for instance. You could have a slime layer or a capsule as well. Here on the left, remember we talked about gram-positive cells, which have a thick layer of peptidoglycan and lipoteichoic acids in the cell wall. So you should know that gram-positive cells, in addition to their plasma membrane, they have a thick layer of peptidoglycan with teichoic and lipoteichoic acids in the cell wall. And do you recall the difference between that and a gram-negative cell wall? Gram-negative cell walls have a plasma membrane, a thin layer of peptidoglycan, and then an outer membrane. Remember, there's a whole nother outer membrane in gram-negative cell walls. The outer membrane has these specialized pore structures called porins. These are protein channels, essentially, which allow substances to go in and out of the cell, called porins. And do you recall that these little tails belong to LPS, lipopolysaccharides? So the outer part of the outer membrane of gram-negative bacterium contain lipopolysaccharides. And these were important to know about because lipopolysaccharides include a lipid portion, which is called lipid A. And lipid A is very toxic to our cells, to our bodies, right? And so when lipid A gets into our system, it can cause all kinds of problems: fever, sepsis, septic shock, even death. And remember that lipid A is found only in gram-negative bacterium, and it is also known as endotoxin. Remember that the cell wall of bacterium is made of peptidoglycan. Peptidoglycan is composed of these. This is NAG and NAM repeating subunits. NAG standing for N-acetylglucosamine, NAM standing for N-acetylmuramic acid, and we have repeating structures of NAG and NAM, NAG and NAM. So there's a chain of these NAG and NAM subunits, which are NAG and NAM are sugars, and attached to them are little peptide chains which cross-link to one another, forming a very strong cell wall. Again, remember the gram-negative cell wall includes lipopolysaccharide, or LPS, which contains that dangerous lipid A protein component called endotoxin. Then there are some non-typical cell walls associated with bacteria. Remember I told you about these cell walls that contain mycolic acids, also known as cord factor. These are very long chain fatty acids, which are waxy. And these waxy substances make a cell acid fast. So when we do the acid fast stain in the lab, you should realize that this helps to distinguish those cells that have mycolic acids in their cell wall and are known as acid-fast bacterium, such as the bacterium that cause tuberculosis or leprosy. Recall that some bacterium don't have a cell wall, and because of this, they would be pleomorphic. They would not have a particular cell shape. So mycoplasmas are such an example. These do not have a cell wall, so they don't have a particular shape. Next, we talked about the cytoplasmic membrane, and I told you that there are many different functions to the cytoplasmic membrane: energy reactions, nutrient processing, synthesis, but also to regulate transport, to be selectively permeable, allowing certain things into the cell versus out of the cell. So the main function of a cell membrane is to be selectively permeable. We talked about the Gram stain technique and how it was developed by a young medical student named Hans Christian Gram in 1884. He did this staining technique and showed that the gram-positive cells would end up with a purple color, while the gram-negative cells would end up with a pinkish red color. And then I told you how this Gram stain technique works. First, the primary stain, crystal violet, is added to the bacterium, which stains both gram-positive and gram-negative bacterium purple. Next, Gram's iodine, which serves as a mordant. And remember that mordants are substances that keep a dye in place. When you add the iodine, it complexes with the crystal violet, forming a crystal violet iodine complex, which lowers the permeability of this dye. So it makes crystal violet harder to wash out of the cells. Next, you add the decolorizer called alcohol. This decolorizer washes the crystal violet iodine complex out of the cell, but it washes it out of gram-negative cells much, much faster than it washes it out of gram-positive cells. Why? Because gram-negative cells have a very thin peptidoglycan cell wall. Gram-positives have a very thick peptidoglycan cell wall, so that impedes the washing out of the crystal violet. The gram-negative cell wall has a thin peptidoglycan layer, and this allows the washing out of the crystal violet iodine complex much easier. So if you do it just right, remember, by not destaining too much or too little, you should get to a place where you've destained the gram-negative cells, but the gram-positives are still color. And next, what do? You add a counter stain, in this case safranin, which is a reddish pink dye. This gets into all the cells, but it really shines on the gram-negative cells, and you can see gram-negative cells in pink and the much darker gram-positive cells in purple. Remember, we talked about the cytoplasm as well. Bacterium obviously have cytoplasm inside of the cells, gelatinous solution contained by the cytoplasmic membrane. This is mainly water. but you have other substances that are floating around in the cytoplasm, including the nucleoid, right? You've got the circularized DNA called the nucleoid. The circularized single DNA. It's a double-strand DNA, but it's a single chromosome of circularized double-strand DNA. That makes up the nucleoid of the bacterial cells. And remember, the nucleoid is floating around in the cytoplasm. What are plasmids? Plasmids are small, double-stranded circles of DNA. They are called extrachromosomal DNA because they are not part of the chromosome. They are outside of the chromosome. However, they kind of function like chromosomes in that they can replicate themselves. They can they contain genes which can be transcribed and translated to form proteins, and they can be shared during conjugation with the sex pilus as well. Though they're also very important in genetic engineering in the lab setting. What are ribosomes? Remember, ribosomes are found in all the domains of life: bacteria, archaea, eukarya. And the big difference, though, is that bacterial and archaeal ribosomes are 70S ribosomes, while eukaryotic ribosomes are a different shape. They're slightly larger, 80S ribosomes. S stands for Svedberg units, having something to do with how fast these structures move in a centrifuge. And lastly, we talked about inclusion bodies, and I told you that inclusion bodies are storage areas inside of the prokaryotic cells. For example, you could have storage bodies for magnetic particles. You could have storage bodies, these inclusion bodies for phosphate or sulfate or anything that the bacterium needs to store for later. And what is the cytoskeleton? The cytoskeleton is made up of polymers, of proteins that give support to the cell and contribute to the cell shape. And do you recall that some bacterium, particularly members of two genus, the Clostridium genus and the Bacillus genus, are capable of forming endospores, which are very resilient, resistant structures that form in response to lack of nutrients and can withstand harsh environmental conditions until favorable environmental conditions return, and then the endospores can germinate. So there are two parts to this life cycle. The vegetative cell is the metabolically active portion of the cell cycle. This is the mother cell, which is growing and dividing. And when, like I said, when the nutrient conditions dip and the conditions become poor, these vegetative cells can form endospores. This is through a process known as sporulation. The endospores are formed, and endospores are a resting condition. They're a defensive structure. Endospores, which look like little seeds, are resistant to heating, to drying, to freezing, to radiation, to chemicals. It's very difficult to destroy these endospores. Okay, and again, what leads to sporulation? What is the stimulus for endospore formation? Depletion of nutrients, especially carbon and nitrogen sources. This can take about 6 to 8 hours to form an endospore. You should know the sporulation cycle, the steps to sporulation of a bacterium. Step 1, the vegetative cell begins to be depleted of nutrients. This is what triggers sporulation. Step 2, chromosome is duplicated, so we copy the nucleoid, copy the DNA. Step 3, the cell is septated into a sporangium and a forespore. The sporangium is going to become dead mother cell. The forespore is going to become the endospore. Step 4, the sporangium engulfs the forespore further development. You see how the sporangium on the right is enveloping the forespore on the left. Step 5, the sporangium begins to actively synthesize endospore layers around the forespore. So here you can see there are actively layers forming around the early endospore. Next, step 6, a cortex and outer layers are deposited. So a cortex is a peptidoglycan layer of the endospore, and then you have the outer coat layers, which are proteins that are protective as well. Step 7, the mature endospore forms. Notice how the vegetative DNA gets degraded. Step 8, free endospore is released. So the endospore frees itself. The endospore is released from the vegetative cell. And then step 9, germination. This is when the endospore grows out back to a vegetative cell because the conditions have improved. Okay. And again, remember, members of the genus Bacillus and Clostridium are the ones that can form endospores, and these can lead to human diseases. Bacillus anthracis, you should know, leads to the disease anthrax. Clostridium tetani to tetanus and lockjaw. Clostridium perfringens, gas gangrene. Clostridium botulinum, botulism. You should know that endospores are intruders where sterility and cleanliness are important. This is why we need to use autoclaves. This is why we need to use incinerators in the lab, as well as flames in the lab. Hospitals must protect against endospores in wounds. And lastly, we touched on archaea. I told you that archaea are more closely related to the domain Eukarya than they are to bacteria because we have a more common ancestor than either does with bacterium. All right, and that is the end of chapter 4. Hopefully that all made sense. Let's take another quick break time with Gizmo and Wicket and come back strong for our final chapter, chapter 5 for exam 1. Hey everyone, welcome back from break time with Gizmo and Wicket. Let's go ahead and finish off this exam 1 review with chapter 5 covering eukaryotic cells and microorganisms. Remember that eukaryotes can qualify as microorganisms as well. There are unicellular eukaryotes known as protozoa, which are a type of protist. And then there are organisms that are unicellular or can be multicellular. These include the fungi and the algae. And then there are organisms that are small, unicellular when they're acquired or when they infect humans, but then become multicellular, such as helminths. Remember, with helminths or worms, usually it's the unicellular egg or larva that is ingested or that gets into your body, and that is what grows into the multicellular worm. This is why we study helminths with microbiology. Remember, there are certain features of eukaryotic cells: a cytoplasmic membrane, a nucleus, mitochondria, endoplasmic reticulum, Golgi apparatus, vacuoles, cytoskeleton, and even a glycocalyx. However, that you do not have chloroplasts in all eukaryotes, right? Chloroplasts are found in special photosynthetic eukaryotes, such as algae, plants, trees, etc. And also, not all eukaryotic groups have cell walls. Plants and fungi can have cell walls, but usually many eukaryotes lack a cell wall. And additionally, locomotor appendages such as flagella or cilia, not all eukaryotes have these types of appendages. So these are more found in certain groups of eukaryotic organisms. Remember that a eukaryote could have a flagella. However, eukaryotic flagella are much thicker than bacterial flagella, much more structurally complex. They are covered by a cell membrane, and they kind of look like this. This is a cross-section of a flagella on the right. Notice that there are these microtubule doublets in a circle. So you have 9 microtubule doublets surrounding 2 single microtubules. And this is known as the 9+2 arrangement, the 9+2 arrangement of microtubules. That's the structure on the inside of a eukaryotic flagella. Now the cilia are similar in overall structure to a eukaryotic flagella. They are shorter, more numerous, and their motility is different. You see, the cilia have a power stroke followed by a recovery stroke, whereas flagella, eukaryotic flagella, do not spin like bacterial flagella. I should have mentioned that back here. Eukaryotic flagella do not spin like prokaryotes, like bacterial flagella. Instead, they beat back and forth, kind of like a fish tail. Okay, and remember that chitin is found in fungi. Chitin is the cell wall material in several fungi, while cellulose is the cell wall material of different types of plants and photosynthetic bacteria. Additionally, eukaryotes possess mitochondria. The mitochondria is the site of cellular respiration. The mitochondria is very interesting because remember I told you in class that the mitochondria are theorized to have been engulfed by other cells, which then became organelles. Remember the evidence for the endosymbiont theory I told you in class, that mitochondria have their own circularized single chromosome, much like bacteria do. They have their own 70S, not 80S ribosomes, like bacteria do. They conduct cellular respiration like bacteria do. They are roughly the same size as bacteria. The genes on this, the gene sequence found in the DNA molecule in mitochondria matches genes of bacteria. The mitochondria themselves divide like bacteria through binary fission. So there's a lot of evidence supporting the fact that mitochondria at one point in history used to be their own creatures, their own bacterial species. And then at some point they were engulfed by an early eukaryotic cell, and then they kind of lived in this mutualistic symbiotic relationship ever since, where the mitochondria specialized in providing ATP to the cell, and the cell specializes in providing sugars to the mitochondria. Isn't that interesting? And the same thing, remember I told you the same phenomenon with the chloroplast. The chloroplast has its own circularized DNA with sequences similar to bacterial genes. It has its own 70S ribosomes for, and remember what ribosomes do. Ribosomes partake in protein synthesis. That's their function. And so the chloroplasts and the mitochondria are theorized to have originated as their own prokaryotic bacterial creatures in history, and they were engulfed and they've lived together in the eukaryotic cells. Remember that the chloroplasts are the photosynthetic organelle in eukaryotic photosynthesizers. We talked about the ribosomes. Ribosomes in eukaryotes are known as 80S ribosomes, while ribosomes in prokaryotes are known as 70S ribosomes. Ribosomes are involved in protein synthesis. Okay, then we talked about the kingdom fungi. There are macroscopic fungi, such as mushrooms, puffballs, gill fungi, and then there are microscopic fungi, molds, and yeasts. Remember that yeast cells have kind of like this. Yeast cells have a semicircular appearance, kind of an oblong to oval shape appearance. It can use asexual or sexual reproduction. Mold, on the other hand, are hyphal. That means filamentous in cell shape. They form long thread-like cells. And you should know that yeasts divide by budding. Remember in class we talked about how yeast can form these asexual buds, which then bud off from the parent cell, leaving behind a bud scar. Fungal nutrition, we should know that fungi are heterotrophic, which means they acquire nutrients from a variety of substrates, namely organic material, by consuming organic material. They are saprobes, which means they obtain their substrates, their nutrition from dead organic material, such as plant material and animal material. It's from dead plant and animal material. Or they could serve as parasites, right? Some fungi are parasites, which means they can feed off the living bodies of animals or plants. Remember, fungi, molds can form a mycelium. What's a mycelium? A woven intertwining mass of hyphae. Remember, hyphae are the filamentous cells of mold. Well, that fuzzy stuff that you see on top of your moldy bread or moldy fruit, that fuzzy mat is known as a mycelium. It is essentially an interwoven mass of hyphae. Septa are cross walls in hyphae. Fungal spores are reproductive bodies of fungi. Don't confuse fungal spores with bacterial endospores. Endospores are a defensive structure formed by just two genus of bacteria, whereas fungal spores are formed by all fungi, and they are for reproductive purposes. They are far less resistant than endospores. Here you can see the life cycle of a typical bread mold known as Rhizopus dolonifer. This is your typical bread mold. The asexual spores disseminate from this cap. The asexual spores land somewhere, and then there's a germination that occurs, which means an outgrowth from the spore. This forms hyphal cells, which form a mycelium. And then you get these aerial mycelium that form, or aerial hyphae that form. And at the top of the aerial hyphae, you form a cap full of spores. These are if it's a cap like this, it's known as a sporangium. And inside you have your sporangiospores, which can repeat the life cycle again. So again, fungal spores should not be confused with the much more resistant non-reproductive bacterial endospores. Do you remember that there's also two forms of asexual spore formation? You have the sporangiospores versus the conidiospores, also known as conidia. Sporangiospores have a sporangium, which is like a cap that encapsulates the spores, and the spores can rupture and leave the cap. They can disseminate from the cap, right? Whereas the conidiospores don't have that cap. They don't have the sporangium. The spores are kind of naked. They're exposed, right? So the conidiospores have exposed spores, such as penicillium, the fungus called penicillium, the fungus called aspergillus. Those have conidiospores, whereas Rhizopus, which I told you about in the previous slide, the bread mold, it forms a sporangium. So different species of fungi can form asexual spores in different ways. Now, fungi can also form sexual spores as well. For example, a bread mold, Rhizopus, can form this sporangiospore to make asexual spore, but it'll form what's known as a zygospore for sexual reproduction. The purpose of sexual reproduction is to increase genetic variability. When you increase genetic variability, it's advantageous to the adaptation survival of the species. Okay. Obviously, fungi can be parasitic, and they can feed off of our bodies and cause an infection. So we need to be aware of that in the hospital setting. Fungi can be beneficial. We use fermentation to produce many different fermented products. We use the antibiotics that are produced by different fungi for medicines. We have many different good uses for fungi. Okay, next let's move on to the protists. Remember, protists these include the algae and the protozoa. There's the subkingdom algae, subkingdom protozoa. What is a protist? Any eukaryotic, unicellular, or colonial organism that lacks true tissues. So let's start with the algae. These are the photosynthetic protists. Here you can see on the left, this is a green algae. And on the right, you can see another type of algae known as diatoms, forming these beautiful structures due to their silica shells, their glass shells. These are photosynthetic. They contain different types of pigment molecules. They produce oxygen for us to breathe. They fix carbon. They take CO2 out of the air, reducing the greenhouse gas. They're also very important. Algae are very important at the bottom of the food chain. They serve as plankton for other organisms to eat. They are the base of the aquatic food web. Moving on to protozoa. Remember, protozoa are single-cell eukaryotes, but these are more animal-like in that they don't photosynthesize. They feed off of organic material. These protozoa are characterized based on their motility. So what type of motility do they possess? Some are non-modal. Some move with amoeboid motility, which means they form pseudopods, fake feet for motility. Some are ciliated, and some are flagellated. They can also have rudimentary structures that are analogous to mouths, digestive system, reproductive tract, or even types of legs. Now, these protozoa can either be free living, these moving around, scavenging dead plant or animal debris, or grazing on bacteria or algae. However, many are parasitic and can lead to human infection and animal infection, even plant infections. These are the ones that live on the fluids of hosts, and they can actively feed on tissues. These are the ones we'd be concerned with for causing disease. So again, there are different styles of locomotion for protozoa, and we classify the protozoa based on their locomotion. Either they don't have locomotion, or they move with pseudopods or false feet. These are the amoeba. They could be flagellated, such as the trypanosoma. They could be ciliated, such as paramecium. And you could see those types of structures here. Now, several protozoa can form two major life stages: the trophozoite or motile feeding stage, and the cyst or dormant resting stage. Here you can see a general life cycle exhibited by those protozoa that have these two life stages. The trophozoite or active feeding stage continues until there is a drying condition or lack of nutrients, in which case the cell rounds up and loses motility, and an early cyst wall forms. Next, the mature cyst, which is the dormant resting stage, forms, And once this cyst forms, the cell is in a resting state until moisture and nutrients are restored, in which case the cyst wall will break open, releasing the new trophocyte, activating the trophocyte again. Okay. Now, in parasitology, we study protozoa, but we also study helminths, which we'll talk about next. And remember, what is a parasite? It's a term used to denote protozoa or helminth pathogens that live and cause disease. And here are some examples of protozoa that can cause disease. Here's some pathogenic flagellated protozoa, Trypanosoma brucei, which causes African sleeping sickness. Please know about how this particular organism causes African sleeping sickness, whereas Trypanosoma cruzi causes Chagas disease, and it's spread by the kissing beetle. By the way, sleeping sickness is spread by the tsetse flies bite, whereas Chagas disease is spread by the kissing beetle's bite. Now, the helminths. Remember, helminths means worms. There are 3 groups of worms to know about. Tapeworms, which are known as cestodes, flukes, which are known as trematodes, and roundworms, which are known as nematodes. Okay, these are very interesting organisms with their organ organs and organ systems. They have a reproductive tract. They have a primitive digestive, excretory, nervous, and muscular system. Thick cuticles for protection, mouth glands for breaking down host tissue. The complete life cycle involves a fertilized egg, a larval stage, and an adult stage. It's usually the fertilized egg or the larval stage during which infection occurs, where the unsuspecting person ingests the egg or the larva, okay, or the egg or larva get into the skin, for instance, and then once inside the body, the larva grow to become the adult, and then the life cycle, continues. Again, how did these worms reproduce? Nematodes, which are also known as roundworms, these have distinct sexes, the male and the female sexes, with different morphologies which do sexual reproduction. Trematodes, such as flukes, these ones have distinct sexes or can be hermaphroditic. Hermaphroditic meaning male and female sex organs on the same worm. These can self-fertilize and produce offspring. And the cestodes, this is a type of tapeworm, or these include the tapeworms, I should say. And these are generally hermaphroditic, again, possessing both the male and female sex organs and can produce offspring by themselves. Now, how do you pick up these worms? You can pick them up through food, soil, water, or other infected animals, usually oral intake by drinking contaminated water or eating contaminated food, or through penetration of the skin. Okay. There's about 50 different species of helminths that parasitize humans. All right, and that about wraps it up for chapter 5. We did it. We got through all the material. I tried to highlight some of the more important points here and help you to focus on some areas which are, more low-hanging fruit for the exam. Hopefully, this was a good little review for you. We basically went through the handouts. What I would do to prepare for the exam is study through your handouts, especially all these terms in bold, all these important terms. Go through, study your handouts several times till you learn all the material. Make sure you got it, and then come in strong for the exam. I think you guys will be set. I wish you the best on the exam. Study hard, and I'll catch you guys next time.
Dr. D, Dr. D, Dr. D. Dr. D, Dr. D, Dr. D. A Dr. D, Dr. D, Dr. D. Dr. D, Dr. D, Dr. D. A Dr. D, Dr. D, Dr. D. Dr. D, Dr. D, Dr. D. A Dr. D, Dr. D, Dr. D. Dr. D, Dr. D, Dr. D.